Hello and welcome dear students. Today we are going to discuss about the topic Hardy-Weinberg principle, applications and exceptions. The main objectives of today's lecture are to know the basics of Hardy-Weinberg equilibrium, to discuss about the applications of Hardy-Weinberg principle, to discuss about complete dominance, to discuss about the sex-linked loci, to discuss about the exceptions to the Hardy-Weinberg equilibrium, to discuss about the Hardy-Weinberg principle and evolutionary implications, and finally to understand the significance of Hardy-Weinberg law or principle. Dear students, let's start with the introduction first. One of the most important principles of the study of genetic composition and difference in populations is the Hardy-Weinberg equilibrium principle. Also described as this principle gives the genetic parameters for a population that is not evolving. In such a population, genetic variation and natural selection do not occur, and the population does not experience change in genotype and allele frequencies from generation to generation. In absence of factors that alter allele frequencies, the Hardy-Weinberg principle shows that Mendelian loci segregating for multiple alleles in diploid populations will maintain predictable levels of genetic diversity. This describes how a population can remain at genetic equilibrium, referred to as the Hardy-Weinberg equilibrium. Genetic equilibrium occurs when there is no evolution within the population. In other words, the frequency of alleles, that is variants of a gene or alternative forms of a gene, will be same from one generation to next generation. At genetic equilibrium, the gene or allele frequencies are stable, they do not change. For example, let's assume that red here is determined by the inheritance of a gene with two alleles R and R. Capital R is dominant whereas small r is recessive. The dominant allele R encodes for non-red here, while the recessive R, that is small r, encodes for red here. If a population's gene pool contains 90% of the capital R and 10% R, that are the small r alleles, then the next generation would also have 90% capital R allele and 10% small r allele. However, this only works under a strict set of conditions. The Hardy-Weinberg principle or law was developed by the mathematician Godfrey Hardy and physician Wilhelm Weinberg in the early 1900s. They constructed a model for predicting genotype and allele frequencies in a non-evolving population. This model is based on 5 main assumptions or conditions that must be met in order for a population to exist in genetic equilibrium. These 5 conditions are as follows. The number 1, that mutations must not occur to introduce new alleles to the population. Second, that no gene flow can occur to increase variability in the gene pool. That a very large population size is required to ensure allele frequency is not changed through genetic drift. Number 4, that mating must be random in the population. And the 5th is that natural selection must not occur to alter gene frequencies. The conditions required for genetic equilibrium are idealized, as we do not see them occurring all at once in nature. As such, evolution does happen in populations. Based on the idealized conditions, Hardy and Weinberg developed an equation for predicting genetic outcomes in a non-evolving population over time. This equation is described as p square plus 2pq plus q square is equal to 1, is also known as the Hardy-Weinberg equilibrium equation. It is useful for comparing change in genotype frequencies in a population with the expected outcomes of a population at genetic equilibrium. In this equation, p square represents the predicted frequency of homozygous dominant individuals in a population, while as 2pq represents the predicted frequency of heterozygous individuals, and the q square represents the predicted frequency of homozygous recessive individuals. That means p represents homozygous dominant, where q represents homozygous recessive. In the development of this equation, Hardy and Weinberg extended established Mendelian genetics principles of inheritance to population genetics. Dear students, let's now discuss the applications of the Hardy-Weinberg principle. Natural populations persistently depict genetic variation altering from mutation, genetic drift, migration, sexual selection, and natural selection. The Hardy-Weinberg equilibrium provides a mathematical criterion of a population that is non-evolving, which can be compared to evolving populations. Over time, if the allele frequencies are noted and estimated for the expected frequencies based on the values of Hardy-Weinberg principle, then workings that drive the evolution of the population can be hypothesized. The law offers a prototype which is typically used as a point of origination to study the population genetics of diploid entities which fulfill the fundamental assumption of random mating, large population, no mutation, migration, or selection. However, the Hardy-Weinberg equilibrium model is not applicable to haploid pathogens. In the event of a population not being found in Hardy-Weinberg equilibrium, one of the assumptions in this law then gets violated. This conveys that selection, non-random mating, or migration has influenced the population, in which case experiments are carried out and hypotheses are advanced in order to understand the reasons behind the non-equilibrium of the population. Genetic variation in natural populations is frequently seen changing as a result of mutation, genetic drift, migration, sexual selection, and natural selection. The Hardy-Weinberg equilibrium offers a formula that distinguishes between populations that aren't evolving and populations that are evolving. The process that underlies population evolution can be hypothesized over time if the allele frequencies are noted and estimated for the predicted frequencies based on the values of the Hardy-Weinberg principle. In order to examine the population genetics of diploid creatures which satisfy the fundamental assumption of random mating, huge population, no mutation, migration, or selection, the law provides a prototype which is often utilized as a point of origination. However, haploid pathogens do not fit the Hardy-Weinberg equilibrium paradigm. One of the presumptions in this law is broken if a population is not determined to be in Hardy-Weinberg equilibrium. This implies that selection, non-random mating, or migration have had an impact on the population. In this situation, tests are conducted and hypotheses are put out in order to determine the causes of the population's lack of equilibrium. Dear students, let's now discuss the complete dominance. Allele frequencies can be detected in the presence of complete dominance when Hardy-Weinberg equilibrium prevails, wherein it is not possible to differentiate between two genotypes. Two genotypes, that is AA, capital A, and capital A, small a, that is homozygous versus heterozygous, have the same phenotype as a result of complete dominance of capital A over a small a, and can help determine the allele frequencies from frequencies of the individuals indicating recessive phenotype small a small a. Here the frequency of small a, that is the recessive allele homozygous one individual, should be equivalent to the square of the frequencies of the recessive allele. The frequency of the recessive alleles AA individuals must be equal to the square of the frequency of recessive allele Q. Let us suppose Q is equal to 0.5, then Q square is equal to 0.5 raised to power 2 is equal to 0.25, that is the 1/4. In other words, when AA or recessive phenotype allele AA is 0.25 in the population, then it follows that the frequency of the recessive allele A, that is small a, is square root of 0.25 multiplied by 0.5. The frequency of the dominant allele A would be 1, that is Q or 1 is equal to 0.5 is equal to 0.75. Dear students, let's now discuss about the multiple alleles. Calculation of genotypic frequencies at a locus with more than 2 alleles is allowed in the Hardy-Weinberg principle, for instance in the ABO blood groups. 3 alleles are present in IA, IB, IC with P, Q and R frequencies respectively, where P plus Q plus R is equal to 1. With random mating, the genotype of population will be given by P plus Q plus R raised power square. Dear students, let's now discuss about the linkage disequilibrium. For example, take 2 or more alleles on the same chromosome at 2 different loci with 2 or more alleles. As a result of genetic exchange by recombination taking place at regular time intervals at 2 syntic loci, the frequency of allelic combinations attains equilibrium. In the event of not being able to attain an equilibrium, alleles are known to be in a linkage disequilibrium, which is as a result of 2 or more linked alleles to be inherited jointly or together more frequently than expected. Such gene groups are also known as supergenes. Dear students, let's now discuss about the frequencies of harmful recessive alleles. The Hardy-Weinberg law can also be used to calculate the frequency of heterozygous carriers of harmful recessive genes. If there are 2 alleles, capital A and small a, at an autosomal locus with frequencies P and Q in the population, and P plus Q is equal to 1, then the frequency of capital A, small a and small a genotypes would be P square plus 2PQ plus Q square. If the AA, that is small a genotype, expresses a harmful phenotype such as cystic fibrosis, then the proportion of affected individuals in the population would be Q square, and the frequency of the heterozygous carriers of the recessive allele would be 2PQ. To simplify this concept, suppose 1 out of 1,000 children is affected with cystic fibrosis. Then the frequency Q square is equal to 0.001, so that Q is equal to root of 0.001, which is about 0.032. Then the 2PQ is equal to 2 multiplied by 0.032 multiplied by 0.968 is equal to 0.062. This means that about 62 individuals out of 1,000 or 1 out of 16 is a carrier of the allele for cystic fibrosis. Because the number of individuals with small a who are actually affected is 1 out of 1,000. This implies that the frequency of heterozygous carriers is much higher than that of affected homozygotes. Similar calculation shows that when an allele is very rare in the population, the proportion of carriers is still much higher and or affected homozygotes much lower. Thus, lower the frequency of an allele, greater the proportion of that allele that exists in the heterozygotes. Dear students, let's now discuss about the sex-linked loci. It is possible to apply Hardy-Weinberg law for calculating gene frequencies in case of sex-linked loci in males and females. Red-green color blindness is a sex-linked recessive trait. Let small r denote the recessive allele which produces affected individuals and capital R the normal allele. The frequency of R, that is capital R, is P, and of small r is Q, where P plus Q is equal to 1. The frequencies of females having capital R, small r, small r genotypes would be P square, 2PQ, Q square respectively. Males are different as they are hemizygous, have only one X chromosome derived from the mother with a single allele, either R capital or small r. The frequency of affected small r males would be the same as the frequency of the small r allele among the X, that is Q. The frequency of normal R, that is capital R males, would be P. Suppose frequency of small r alleles is 0.08, then the incidence of affected males would be 0.08 or about 8%. The frequency of affected small r females would be 0.08 raised power 2 is equal to 0.0064 or 0.64%. Thus, the Hardy-Weinberg law explains that males would be affected 100 times more frequently than females. This is actually what is observed. Males are more affected by sex-linked recessive traits than females. The difference between the sexes is even more pronounced if the recessive allele is still more rare. The incidence of a common form of hemophilia is 1 in 1,000 males, thus Q is equal to 0.001. However, only 1 in 100,000 females will be affected. Thus, males could have hemophilia 1,000 times more often than females. So, dear students, let's now discuss the exceptions to the Hardy-Weinberg equilibrium. Processes that alter patterns in genetic variations are: the first one is mutation. It has a mild impact on the allele frequencies. Even if there persists a sturdy selection against the alleles in the population, recurrent mutations will conserve it. Mutation in only one direction can cause one allele slowly to replace another. Mutations in both directions results in an equilibrium with frequencies determined by the mutation rates. Mutations could lead to occurrence of new alleles and thereby it changes the gene pool of the population. It may be favorable or deleterious to the individual's ability to survive. If changes are advantageous, then the new alleles will tend to prevail by being selected in the population. One of the requirements for a population to be in a Hardy-Weinberg equilibrium is that there be no mutations. These alterations affect the genes and alleles that make up the genetic diversity of a population. Dear students, the second one is natural selection. Differential fitness of individuals in a population based on differences in phenotype. Typically, this leads to a change in the allele frequencies and is a rapid one. There are few types of selections. The selected ones can result in equilibrium with no loss of alleles, namely balancing selection, while some other selections such as directional selection can gradually result in the loss of alleles. Selection occurs whenever different genotypes contribute genes unequally to the next generation. This is the most important and most frequent departure from equilibrium conditions. Size of the population being small can lead to a random alteration in the allele frequencies, which can be attributed to the sampling effect known as genetic drift. When alleles are found in a fewer copy, sampling effects are significant. The third one is migration. Two or more than two populations can be associated together genetically with migration. Here, amongst the populations, the allele frequencies have the tendency to become more homozygous. Essentially, a few migration models are the Wallund effect, that is non-random mating. Hardy-Weinberg proportions typically are invalid for such models. Migration between populations always causes the gene frequencies of the receiving population to shift towards those of the immigrants. Non-random associative mating. When individuals choose mates non-randomly based on phenotypes, for example, female mate choice. Positive associative mating when mates are more similar to each other than expected by chance. The homozygote frequency increases in this case. Negative associative mating when mates are less similar than expected by chance. Here the heterozygote frequency increases. It is less common in nature. Another one factor is the genetic drift. It is the change in allele frequency due to random chance. It's associated with sexual reproduction and is more common in small populations. Genetic drift happens due to the sampling error. If the population is not large, gene frequencies can fluctuate randomly in either direction simply by chance. Dear students, let's now discuss about Hardy-Weinberg principle and evolutionary implications. The Hardy-Weinberg principle demonstrates that Mendelian loci segregating for multiple alleles in diploid populations may maintain predictable levels of genetic diversity in the absence of influences that alter allele frequencies. These predictions are frequently visualized by plotting p square, 2pq, and q square as a function of allele frequencies. This graphical depiction highlights two key consequences of the Hardy-Weinberg principle. When p is equal to q is equal to 0.5, the population heterozygosity, that is frequency of heterozygotes, is highest. Given that when q is close to 0, q square is considerably less than 2pq, and when p is close to 0, p square is much smaller than 2pq. Rear alleles are predominantly found in heterozygotes, as they must be. The second assertion acquires special significance when we take into account how natural selection can influence the frequencies of novel mutations. All individuals in a population must be homozygous for a beneficial allele for that allele in order for it to exist if the other Hardy-Weinberg law assumptions are true. The initial increase in the frequency of a common favorable dominant allele is greater than that of a rare advantageous recessive gene. Because uncommon alleles are frequently found in heterozygotes, as we have seen, the natural selection won't detect a novel recessive mutation until it occurs in homozygotes with a frequency high enough, perhaps through drift in a genuine finite population. However, because a new dominant mutation affects fitness in heterozygotes, natural selection can detect it right away. Even though Hardy-Weinberg in 1908 demonstrated that dominance alone does not change allele frequencies at a site, dominance interactions between alleles can have a significant impact on evolutionary trajectories. Selection, mutation, migration, and genetic drift are the processes that alter allele frequencies. When one or more of these processes is active, the population deviates from Hardy-Weinberg's assumptions and evolution takes place. Dear students, let's now discuss about the significance of the Hardy-Weinberg law. The significance of Hardy-Weinberg law is that the model helps to evaluate the real genetic structure of a population over time. Only with the genetic composition we predict if the population was in equilibrium with Hardy-Weinberg that's not evolving. In evaluating the genetic variation present in a population, Hardy-Weinberg law is essential and compares the real variation to the calculated value of Hardy-Weinberg law if the population was in equilibrium. If the real frequency in a population varies from the predicted value, then it is an indicator of one or more assumptions being disrupted and violated, which can be further investigated. It also helps in predicting the prevalence of a negative recessive gene in the heterozygous carriers. Dear students, let's now conclude the lecture. The Hardy-Weinberg model states that a population will remain at genetic equilibrium as long as 5 conditions are met: no change in the DNA sequence, no migration, a very large population size, random mating, and no natural selection are the 5 principles of Hardy-Weinberg principle. The Hardy-Weinberg law is applicable when there is random mating. Random mating occurs in a population when the probability of mating between individuals is independent of their genetic constitution. Such a population is said to be panmictic or to undergo panmixis. The Hardy-Weinberg model also serves as mathematical formula used to predict allele frequencies in a population at genetic equilibrium. If we know the allele frequencies of one generation, we can use this formula to predict the next generation. Again, this only works if all 5 conditions are being met in a population. Dear students, it was all about today's lecture regarding Hardy-Weinberg principle, applications and exceptions. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.